Okay, good morning, everyone. My name is Paulina Martinez, and I'm coordinating the work on Turbot in the AQUA-FAANG project. So, Turbot is a species, a flatfish species, which main production problems are related with diseases, but growth is a target of breeding programs, and also sex control is important because the females growth much higher than males, and the production currently, this is from the last week, 100,000 tons. China is the biggest production, but Spain is the second one. It has a big, high commercial value, and the companies are in the 6th generation of the breeding programs. Within this project, in collaboration with an MBU and the 6th group, we have assembled the Turbot genome and identified the sex-determining gene. That was very important to have a very robust genome and well-assembled, terrific genome, as Sig named when it was assembled. And that is being very easy to manage, as the EBI people are seeing, because it has no duplication. It's a very compact genome. We have very important previous information in the Turbot regarding the QTLs with the different traits we were working with, resistance to pathologies mostly, but also growth. And Oscar, in his PhD thesis, he has reannotated and updated all the information previously released in the new Turbot genome. That is very important, because 20% of the information previously annotated in the genomes was reannotated and was relocated and remapped. Oscar identified common genes to the different pathogens, suggestive of general immunity of the fish, but also specific genes related with the viruses, bacteria, or parasites. So this is very important baseline for working for the future identification of allelic variants related with elements. So this has been published within this project in the thesis Oscar, with integration of the host pathogen functional genomics in the chromosome level genome of Turbot. So going ahead with the work done in this project regarding the atlas, as has been said, we developed a depth map using the stages that Matthew told before. We also analyzed 6 organs in juveniles and adults, males and females. And we also developed an immunomap that will be presented tomorrow by Oscar. A paper is being submitted for publication very, very quickly. In addition, in collaboration with Dan's group and Diego's group, 54 breeders of the main companies of a broodstock, representative of the main companies of Turbot, were resequenced at the whole genome level, and using the previous information of RADSEC, in imputation, a lot of information was gathered regarding the SNP variants across the whole Turbot genome and structural variants across the whole Turbot genome. So we used transcriptomics, epigenomics, ATAC-seq, ChIP-seq, that was previously presented by Matthew, and the bioinformatic tools generated by the BI. And we used a particular, our homemade pipeline for microRNA annotation. We have used previously PROST with Senegal Soul, as Julian knows very well. We have a talk in Granada. And we used a new pipeline using short stacks to identify the significant clusters, and then the most frequent sequence to be blasted or to be compared with the FISMIR database, taking the harping we blasted against the Turbot genome, and then counted the number of mature 5P and 3P. 362 libraries were constructed for a body map. The main problem was related with brain and liver, two fatty organs that made some problems for doing that, but mostly of the libraries for seeing were produced. And with the maps, 102 libraries were produced. In this case, the main problem came from the very hard chorion of marine fishes, but all the libraries were produced as foreseen in the project. So with the raw data coming from the sequencing data, we used DiffBind to identify the significant peaks for attack in the four chipsack marks. Results seems quite reasonable because the attack peaks were double for all tissues. We analyzed down most of the chipsack peaks, and there was some variation. For example, the brain in the female was very low, as in the gonad, but it is quite consistently across all the as foreseen. So then we used this information to look for the correlation using, in this case, deep tools, to look for the correlation of the different techniques and samples across the whole genome using 1 kilobase window. And we produced a heat map of this correlation. The clusters were mostly related with the technique used. For example, this cluster is related with RNA-seq, this cluster is related with attack-seq, and this cluster is related with the silencer signal, and a mixture of clusters were detected with the other marks related with promoters, enhancers, and active genes. Regarding the tissues, the tissues clustered within each technique. For example, here in RNA-seq, each tissue has a specific cluster, but it's interesting that the muscle and the gonad conformed a cluster, a huge cluster that suggests some similarity between them. We don't know exactly why. We performed the same analysis for the maps, using, again, the DiffBind and deep tools. The results were very consistently in both cases. So we then moved to categorize the chromatin state using chromatography, and we identified in the case of body map, 10 different chromatin states, and these chromatin states were grouped in 5 different features: promoters, enhancers, repressed, attack islands, and quiescent. And with this information, this is a representative of the gill female. We detected some differences between the different tissues, especially in the gonad, the muscle, and the head kidney. The information we have this information available if needed. After this classification, we identify the number of quiescent, attack islands, promoters, enhancers, and repressed regions for the different tissues. For example, in the case of brain, a huge repression signal comparing to the other tissues. Quiescent was very common, for example, in the gonad and also in several other tissues. And this information was also generated for the maps. We categorized the chromatin states with chromatography. In this case, very consistent across the different developmental stages. And again, we categorized 5 different features across the genome. And as in the body maps, we presented the histogram with the different types of regulatory elements across the different stages. So then we identified active enhancers across the genome using crossing the information with the attack islands as previously was presented by the EBI people. And we identified clusters of enhancers across the whole genome. You see here 2 clusters of enhancers in chromosome 1. As an example, in chromosome 1, we detected 64 clusters of enhancers with a mean and average difference between consecutive enhancers of 2 kilobases and representing approximately 5% of the whole chromosome length. This information, the clusters of enhancers are very interesting because it's supposed that are very conserved across the evolution. So Juan, which is presenting, is initiating the work with the comparative regulomics in flat fish, will use this information to check the conservation of these enhancers, clusters of enhancers across the different species. Then, within the active enhancers, using Homer, we identified the motifs of the most significant transcription factors in the different, for example, in the case of the MAPS, Pharyngula, the 2 first transcription factor motifs identified were RFX and this one. You will see here that in the different stages of the MAPS, the SOX genes, the SOX transcription factors are very important and apparently very specific of the different stages. And we performed the same work for body maps. I have no time to explain this. In this case, it was very clear the high specificity of the different transcription factors regarding the tissue, excluding the muscle and the gonad that apparently shared the same transcription factors. Then we, using the information of the small RNA libraries, we identified within the pipeline around 250 microRNAs and 52 pWRNAs. We mapped in the Turbot genome this in black, the microRNAs, some clusters of microRNAs are detected, and in red, the pBRNAs. You see that the pBRNAs clustered very clearly across the Turbot genome. And we used the information of the number of reads for each of microRNA, and gathering all the information, we can see that the microRNA are practically inexistent at the beginning of the development, but is increasing across the developmental stages. And regarding the tissues, depending on the tissue, the activity of the microRNAs is very different. For example, apparently no activation of microRNAs in the gonad, but high activation, for example, in the brain. We use a PCA to compare all this information across all samples we analyzed in the project, and we could separate clearly using the microRNA information, the embryonic stages from the body map organs across the first and the second dimensions. The most important microRNAs separating the tissues across the first dimension was there, and across the second dimension was this. And in the body map, you can see here that the brain clearly separates from the other tissues, and the second component separates the other tissues of the body maps very clearly. So variants in the regulatory elements, after identifying active enhancers, Cristina and Joe from the Diego Santana's group identified more than 1 million of SNPs across the Turbot genome, and overlapping with the enhancers 65,000. And in the case of Joe, he identified very consistent 9,000, closely to 9,000 structural variants, duplications or deletions, and 292 overlapped with the active enhancers. So we crossed the information. This is the representation of the information crossing the active enhancers with in this case, the structural variants. For example, this enhancer that starts here, is in chromosome 1, and then here, this is a deletion. This is the position of the initiation of the deletion, the ensemble annotation of the gene, and the chromatin and the chromatin categorization of enhancers and promoters. This is a figure done by Joe, a particular gene where enhancers and promoter apparently are crossed by a particular structural variant. So finally, application for Turbot industry. We have developed 3 years ago a multi-species in chip. This is very important for a species like Turbot that has not very big production. So this reduces the price very much, for 20 euros currently per chip. We are combining whole genome sequencing in the parents with imputation after genotyping with the chip of the offspring. Genomic selection programs are currently being going ahead with Turbot farms, and the simulation studies performed by the Diego's group suggest that this strategy is good for working with breeding programs. So this is the first chip we produced 2 years ago. We based on the polymorphism, the homogeneous distribution, no other polymorphism. Additionally, in Turbot, we included markers related with sex, size, resistance, geographic and farm markers, in association with environmental variables. In November of this year, we are releasing a second multi-species chip that includes rainbow trout and other mollusc species. And for 2025, we expected to include the new information coming from this project in the new Turbot chip, in the new multi-species chip. And Adrian will have a presentation of this application for the Turbot breeding programs tomorrow, I guess, or no, on Friday, sorry. So the take-home messages: an atlas was constructed, the results seem to be consistent, a total number of ataxic, epigenetic marks characterizing active enhancers and promoters, transcription factor motifs identified, microRNAs and PBRNAs identified, a huge amount of structural variants and SNPs overlapping with, some of them overlapping with the active enhancers. And this information is being evaluated to check the increasing accuracy of breeding values in breeding programs. So I want to thank to all the people from the AQUA-FAANG project, the Rolling Institute, the groups of Dan and Diego, of course, the sixth group. I don't have the picture, sorry. And it disappeared, but my group was also involved in this work, you know? And many people from my group. So that's all.